Okay. So I'm going to break up my talk into two sections. As advised by the organizers, I would devote the first half to some fundamental concepts in microbiome research and background. And then in the second half of my talk, I will elaborate more about an emerging area where we are studying how a minor component of the gut microbiota may have a big influence on the larger bacterial community. So if I may just get my pointer out. Okay. So yes, Wendell is correct. We have known each other for many years. And this is just a more recent pre-pandemic picture of Wendell visiting my lab several years ago. And what's common for the two of us is we have a common student, right? So John Yason pursued his PhD and postdoc with me, and I'll be sharing some of the data that he produced in this talk. And I come to understand that now he's an associate dean in the university. So he's risen very quickly since working on Blastocystis. So Blastocystis is a good parasite to work on. So let me just go back to the concept of the microbiome, microbiota, and the gut. And maybe I'll start with a question, you know, when I talk about humanity or being human, would you include, besides your own tissues and your organs, would you include your bacterial component or the microbiome or microbiota component in and on your body? And many people would say no, right? I don't, these are external to me. They don't make me who I am. But in experiments where either humans or mice are grown or developing in the absence of their resident microbiota, there are very clear physiological deficits in their development. And quite starkly, the immune system and their neurobiology is all compromised in the absence of microbiota coevolution. So it seems that we have to be considered together with our microbiota when we talk about humanity or being human. So what are the terms that are often associated with microbiome research? So we tend to use the term microbiota to refer to the ecological community of mainly commensalic, symbiotic, and sometimes pathogenic microorganisms in a particular niche, right? So it could be the microbiota on my mobile phone, for example. So this could be microbiota on a stone in a pond in a forest. But in the context of medical microbiology, we're talking about microbiota on our skin surfaces or in our gut, for example. Microbiome is different. It's the collection of genes or genomes that make up your microbiota. But sometimes these terms are used interchangeably, all right? But strictly speaking, one is the organisms, the other one is the genes and genomes. Metagenomics is the study of all the genes, right, within that particular microbiota. And with the advent of high throughput sequences and high capability sequences, this can be done very easily and very quickly. Another term that we use as opposed to homeostasis, where you have a very balanced gut ecosystem or microbiome ecosystem on your tissues or in your body, as opposed to that, we have this term called dysbiosis, which is just alluding to imbalance in the composition of the microorganisms in that particular niche. Okay, and then the fifth term that's often used is operational taxonomy unit, and these are the kind of identifiers that arise from your sequences. So it could be species, genus, class, order, or family, depending on how deep you sequence or how much information you get. But in general, we just call them OTUs, right, for a simpler definition. Now, in any study on health and microbiome or disease and microbiome, we look at often what is increased or decreased in a particular in a particular niche. So in this example, we have certain groups of bacteria that are increased in inflammatory bowel disease, whereas there are other groups that are decreased. So this is a very simplistic overview, right? What are the tools we use for the research? Now, the beauty about sequencing tissues from a particular niche is that you do not need to culture, and culturing actually is a challenge in most circumstances, because once you culture, the populations start to show differences in their abundances and properties, right? Molecular analysis is almost always done. At the most basic level, you can do qPCR on specific targets, but this is limited, although in John's paper that I'll talk about later, there is a lot of value in doing that. Most often, and because the cost has gone down significantly, we can do 16S ribosomal RNA sequencing of the prokaryotic population, right? And some groups with bigger grants, not my group, but other bigger consortia and all that, they can do whole genome sequencing because the cost is quite, can be prohibitive, but because of whole genome sequencing, as in contrast to 16S, you can get actually much more information. So beyond species and genus, which is more 16S sort of associated, you can get information on polymorphisms, variants, and importantly, because you're getting the genomes of the prokaryotes, you get functional information as well. But for all this, it's really not useful unless you have the computational tools, because ultimately you're going to crunch the data to provide useful information. Therefore, in any microbiome group, you need a bioinformatician. You need someone who knows bioinformatics to analyze the data and give you objective, relevant information. Right, and the first, I guess, term that you would read in any microbiome paper is the term alpha diversity. And this essentially means what you're asking the question, how many organisms are there in your sample? So how rich is your sample? And this term is not a new term. It's a very old term used in ecology. So when you study, again, a forest or a piece of land, you're asking how many organisms there are, how many animals there are in this particular niche. But it's been recently adapted for use in microbiome studies, right? So in the context of the gut microbiome, you're actually asking the question, how many bacteria species are there in this particular in the gut? Okay, and at the most basic level, you ask the question, how many OTUs are there? Okay, remember the term I use is the identifier for your 16S sequences, and you just count the number of species or OTUs. But there's another component that's quite useful in microbiome studies, and that is how are the abundances of your bacteria distributed? Are they very even? Do you have roughly same numbers of certain dominant species, or is there an overgrowth of a particular species? So evenness also can be a determinant of health or disease. So if there's a loss of evenness, then it may suggest a less healthy situation. Okay, and these are usually represented as box graphs, and normally you will see three readouts: richness, Shannon, or Simpson index. And I won't go into the details, but there are certain considerations in the formula for these different indices. For example, Shannon index takes into account the richness and the evenness, but assumes that all species, abundant or rare, are important, whereas Simpson takes into account dominant species. Okay, so here's an example of a data that was produced recently in my lab by Teng Lei, my former PhD student, and he's looking at Blastocystis colonization in sick animals, animals that have colitis. So on day one, if you look at the richness index, on day one, for all the controls, with or without Blastocystis, they have a certain number of species, so about 200 species, right? But after they have colitis induced by a drug, the species richness drops. But if they have Blastocystis, they are able to retain their diversity, right? And in terms of evenness, for Simpson and Shannon, it's even more stark. So this is how we measure alpha diversity. Now, another important term when we work on microbiome studies is this term called rarefaction index. And essentially, you're asking the question, how far have you sequenced your samples? Do you have the representative number of species through your sequencing? And if you don't sequence far or deep enough, you won't get representation. So as the more you sequence, the more unique identities you pick up. And at some point, you reach saturation. And at this point, you can then say these samples are representative of the composition of the niche, and I can use this for further analysis. So for example, in this particular sample, and this is a reflection of the samples I showed before, this has much fewer species than these other samples. And this is, again, the disease state, right? The state by which inflammation or colitis has been induced and the number of species has dropped. Whereas this level here, which has the most species, is the one that has been colonized with a particular subtype of Blastocystis. So just to recap and give some kind of a pictorial understanding of alpha diversity, you can have communities, and each community you have a certain number of species. Here you have 5 different species, right? Represented by colors, and in community 2 you also have 5 different species, but community 2 has less evenness because there's the overgrowth of a particular blue community. So while the OTU numbers are the same, their Simpson and Shannon indices will be quite different because evenness is factored into these calculations. And this is just a rarefaction index kind of a point that you have to sequence deep enough, and then you can compare the difference between the richness or the OTU numbers between one sample over the other. Okay, so let's kick it up a notch and talk about another ecological term. And while alpha diversity talks about number of species or organisms in one sample, beta diversity is a measure of similarity or dissimilarity between 2 communities. So this is useful for your control community versus your test community. And again, this is not a new term. It is a very old term used in ecology, right? But it's very useful for determining compositional changes. So among the species in one community, what is common in the 2 or what is unique in one over the other. And this is usually represented as a dot plot, or we call this a principal coordinate analysis or PCOA plot, where in essence, you're using a formula to determine how dissimilar are 2 communities, right? You're measuring dissimilarity, and the higher the index, the more dissimilar are 2 communities. So again, I think bring you back to the same experiment where you have a bunch of controls untreated before they are induced to be sick, and they all cluster in one on the left part of the graph, right? And over the course of time, when you induce colitis, the control shifts significantly to one position of the graph, whereas the Blastocystis colonized mice have shifted to a slightly different location, right? And from there, you can get compositional changes as well. Here, again, the 3 columns are all the controls, so they don't show much difference in terms of their species abundance. But if you look at the Blastocystis conditioned or colonized context, you have certain bacteria that are either over or underrepresented. For example, Odoribacter or Clostridia have much higher abundances in these conditions than in the controls. Right. So that's kind of the basic understanding of beta and alpha diversity. And now let's extend it to a very famous study that took place, that was published a decade ago. And this is Pierre Bock's group, and he's a big microbiome guy from Germany. So they're part of the EMBL study group. And they proposed something quite provocative in this Nature paper, right? Now, of course, for Nature, it has to be big and provocative. And what they proposed is that no matter who you are, where you're from, you have 3 types of bacteria in your gut. Only 3 types. It doesn't matter how old you are, which country you're from, and all that. So they had 33 samples of a European cohort, and they did the beta diversity of the gut, and they found that, oh my goodness, you're either one of 3 enterotypes, right? Either Bacteroides, this one is the Ruminococcus or Prevotella, right? And when they sequence, subsequent studies seem to support that, right? And for all these cohorts, there's one particular dominant type of bacteria. So for the Bacteroides enterotype, there's more Bacteroides, and subsequently for the Prevotella and Ruminococcus enterotypes, there are more of such bacteria, right? So it's quite provocative because it seems to be independent of age, gender, body weight, and geography, but may be associated with diet. So there's some supposition that Bacteroides enterotypes are associated with maybe a fatty Western diet, whereas for the Prevotella group, it's more linked to more agrarian or vegetable or Mediterranean kind of diet. But recent studies have shown that it's probably not so clear-cut. Depends on how you analyze the data, you actually get them all converging in the middle, right? So it's not, it's provocative, but people are moving away from this rather sensational paper that was published about 10 years ago. So that's kind of the basic kind of layout of the microbiome field. But there are still pressing questions in the community, despite microbiome over the last 10 years becoming a very big and hot topic. The question is, what is really a healthy gut microbiome, right? We always say that if you're diverse, you're even in your microbiome, then you're healthy. But that's not true. We know people who have a so-called healthy microbiome, but who are sick, who have cancer, for example, or who have metabolic diseases and so on. However, what is known is that if you have low diversity and low evenness, then it's likely an indicator of poor health, right? And then a lot of these studies, which I've mentioned, are associative studies. You take a population, you study their gut, then you say, oh, if you have IBS or IBD, then you have this bacteria or that bacteria. These are all association studies, but can we move towards causality studies? And in order to do that, you need experimental animals, right? So in this context, if you ask the question whether parasites or protozoa cause a change in a particular bacterial cohort and so on, then you can induce or colonize with the bacteria, with the parasite, and then look for changes subsequently. And to go back to the title of my talk, what is actually the role of this minor component, the minority of your microbiota are actually eukaryotes. The vast majority are prokaryotes, but it is increasingly clear that there's a vocal minority. So just because they are fewer in number doesn't mean their effects are limited. They can amplify the effects through several mechanisms. So that's kind of the question that we're asking in the laboratory. So let me introduce a parasite that is no stranger to many of you, especially to members of Windell's lab. So Blastocystis has been recognized now as the most common eukaryotic microbe in the human intestinal tract, estimated to be about 1 billion people harboring this organism worldwide. Now, if you take any prevalence study, take any year, so I took 2014 as an example, most of the studies would indicate Blastocystis as the dominant parasite surveyed among populations. It could be in Australia, it could be in Morocco, it could be in Nicaragua. They're all, Blastocystis is very common. And one reason why it could be common is because actually it's made up of 9 or more subtypes. These are genetically distinct types that could even be different species. The clinical significance has been ambiguous for several years, and it's becoming clear now that some subtypes may actually be associated with health and some with disease. And this is the data I'll be presenting with you today. Now, when it's associated with disease, and this is very historical, it's associated with abdominal pain, diarrhea, bloating, and nausea. But as I mentioned, it's of emerging interest as a component of the gut microbiota. So there are, in nature, 17 subtypes, because animals can harbor certain subtypes, but 1 to 9 are human in nature, or human related. And I'm going to talk about 2 subtypes that we have in our lab, and one tends to be more Asia-centric. Subtype 7, common in avian hosts, in ducks, geese, and chickens, and rare in humans globally, but seems to be common in Asia, right? And a recent study in Singapore, we found that the majority of diarrhea patients from our university hospital harbor this subtype. Whereas subtype 4, and the reservoir for subtype 4 is rodents, so your rats, but not mice, strangely, is also common among humans in Europe. And for subtype 4, it's actually associated with a healthy gut microbiota. So we're trying to understand, is there a subtype-dependent linkage between the two? Now, this is a study that John conducted several years back as a fun project, because we had this very cool imaging equipment, but it helped us to determine certain phenotypic differences between the subtypes. So 4 and 7, actually, while they're called blastocystis, are quite different. Subtype 4 is known to be smaller in size, as you can see here. Subtype 7 is larger, right? So there are clear phenotypic differences, even though they're all under the Blastocystis family. Now, so I like to go back to the context of what we call the homeostatic state in the gut and the dysbiotic state. So normally when it's homeostatic, means everything's in balance. You have a nice obligate to facultative anaerobe ratio, and there's a diverse microbiota leading to functional capacity. So if you have more genes, more microbiotas, then you have more a wider array of maybe metabolic and physiological functions that your microbiota can provide to the host, right? Whereas if you've lost the diversity, you have a more simple, less rich microbiota, then there's less metabolic function leading to dysbiosis. For example, when there's overgrowth of a particular pathogen, that can lead to a dysbiotic state and a disease state. So that's the oversimplified idea of how a dysbiotic gut can contribute to disease. But what's the interest in studying Blastocystis and the gut microbiome? Well, there's a call to complete the picture. We're talking about bacteria most of the time, but even though it's a small component, the eukaryotome has been recognized to have a potentially big impact on the entire microbiota and the host physiology. There are some studies that suggest Blastocystis infection promotes dysbiosis and could lead to disease states, whereas a majority of studies show that Blastocystis is associated with healthy individuals with higher gut microbiome alpha diversity. So the reports are conflicting. So why is there such controversy, and is there a way to resolve it? So interestingly, I attended a meeting to me in a very exotic location. I've never been to South America. So there was a Blastocystis meeting in Colombia. And that's where I met Dr. Raul Tito, who at that time, at the meeting time, he published a really big paper where he studied Blastocystis subtypes in a human population in a very large scale population study. And what this paper published in Gut concluded is that microbiota richness and diversity are associated with Blastocystis prevalence and subtype variation. So you have Blastocystis, you carry Blastocystis, you have so-called a more healthy gut, right? But interestingly, because he had so many samples, he could show that different subtypes correlate differently with certain bacteria. So there is a well-known beneficial bacteria called Akkermansia. There was an inverse correlation with Akkermansia abundance, whether you have subtype, depending on whether you have subtype 3 or subtype 4. So the more subtype 3 you have, the less Akkermansia you have, and vice versa for subtype 4. So he concluded that subtypes are important for assessing the role of Blastocystis microbiota host study. So these are just some figures from his paper. So he took 3 cohorts to study, right? The Flemish or Belgian cohort, the American, and then the UK cohort. And just to show you that there are many subtypes in the cohort, but subtype 4 is dominant in the West, right? Subtype 4 is the one in blue here. So it's the dominant subtype, whereas subtype 1, 2, or 3 will be more dominant in Asia, with subtype 7 being more represented. And if you look at the samples in terms of abundance or Blastocystis per milligram of stools, subtype 4 tends to dominate over the other subtypes. So subtype 4 seems to be kind of a commensal of the European or Western population. And if now you go to the beta diversity PCOA plots, which I talked about before, and you group them into enterotypes, right? So if you do a beta diversity plot, you can see now the enterotypes don't link so well together, but you can still differentiate them by colors. So Bacteroides is this group here, Prevotella is this upper group, and Prevotella is on the left, right? And Ruminococcus is on the top. Now you ask the question, for these different so-called enterotype individuals, which one has Blastocystis? Blastocystis tends to go to the Prevotella Ruminococcus groups, and much less in the Bacteroides group. And again, if you talk about health and disease, it is suggested that the Bacteroides group is more linked to Western diet and therefore may be a bit more unhealthy, right? So Blastocystis seems to be found or be associated with the Ruminococcus and Prevotella enterotypes, which are supposedly people with better dietary habits. Okay, so interesting and seems to be linked to health. But here is where the differences start to show. If you look at the correlation between Akkermansia abundance and subtypes, when you have subtype 4, you have high abundance of Akkermansia, subtype 3, low abundance, right? And not so much association with 1 and 2. If you look at abundance of Blastocystis, more subtype 3, less Akkermansia, more subtype 4, more Akkermansia. So there is actually a correlation between more of one subtype and more of Akkermansia. So it's interesting because Akkermansia is healthy, subtype 3 tends to diminish this healthy bacteria. So there may be a slight negative role of subtype 3 in the human population, right? So these are all human studies and associative studies. What about causality, right? Because these are only associated, blasto is associated with this particular phenotype. So in vitro and in vivo studies, which we do in our laboratory, can provide deeper insights. So with in vitro, you can have mechanistic insights. With in vivo infections, where you can introduce the organism through experimental infection, you can determine the causal nature of the interactions. Let me check my time. Okay, my time is okay. So, for example, when you have subtype 4 and you have Akkermansia, it may just be a correlation. There may be some other factor giving causality. For example, if on a hot day, more people buy ice cream, more people get sunburn. But that doesn't mean that ice cream causes sunburn or sunburn causes ice cream, right? It's just an association. So it's in the same way with animal infections, we can move towards causality. Right, so what I'm going to share with you today is studying 2 subtypes, 7 and 4. 7 is John's study, more in vitro and some in vivo work, whereas subtype 4 is a heavily in vivo work done by Teng Lei, my PhD student who's just joined me as a postdoc. And the merits of the studies I mentioned is that while rats have a lot of Blastocystis, mice are negative, are not natural hosts of Blastocystis. So there's no chance that you have a contaminated or immune mouse, right? So for your infection studies, they are clearly naive for Blastocystis. And you can move towards causality, and we can induce illness in the mice to ask the question, does Blastocystis exacerbate the illness or protect against the illness? And I'm alluding to some of the data in the earlier parts of the talk. Now, so I'd like to start with a study that started even before, obviously before we published in 2019. And this is a picture taken when John won a nice award for just the beginnings of the study, together with my former postdoc, Sitara Ajampur, whose work I will describe as well. But this is a very, very big paper in our field, a seminal paper, because not just because of the impact factor, but because it really is the first causal paper showing that subtype 7 can impact the microbiome negatively, right? Not just association. So the study started off very simply, asking a very simple question, and I guess I would say it's an elegant study. You don't need a lot of fancy equipment. It's just counting, right? So what's the question is, how does Blastocystis impact growth of gut bacteria and vice versa? And what John did is just take Blastocystis on its own, representative gut bacteria on its own, or mix them up and then count. For Blastocystis, you need the hemocytometer. For bacteria, you need the drop plate on agar, right? It's a busy slide, but essentially what, I'll just summarize the take-home message. For subtype 7, either isolate B or H. If you look at Blastocystis co-incubated with various representatives of the gut microbiota, most of them, if not all of them, help blasto to grow better, right? They all grow better than the control. And true enough, or similarly, for the bacteria, the 6 bacteria, most of them also grow better in the presence of Blastocystis. So there appears to be, at least in vitro, a symbiotic relationship, save for one, right? So there was an outlier. Bifidobacterium longum, a well-known probiotic and beneficial bacteria, suffers in the presence of Blastocystis. Even here, you can see control versus treated with blasto, there is a significant drop. And blasto seems to do much better in the presence of Bifidobacterium here, right? So the question now is, what's really happening? And because Bifidobacteria are strict anaerobes, and we know that other members of the gut microbiota could be facultative anaerobes, they could exert some oxidative stress on the gut bacteria. So on Bifidobacterium, right? So here we threw in E. coli in a 3-component system. And we asked the question, what do Blastocystis and E. coli do to Bifidobacterium? So here's the result, right? So we asked the question first on E. coli. For E. coli on its own, there's a certain number after incubation, but E. coli does better with B. longum and does even better with B. longum and subtype 7, right? Now, if you look at B. longum, it really suffers from just by giving it E. coli. And if you throw in Blastocystis, there's almost obliteration of Bifidobacterium longum. It's as though these two are ganging up to bully Bifidobacterium longum. So is it oxidative stress? So this question can be answered quite easily by using this oxidative stress markers, right? So commercially, you can buy these dichlorofluorocene diacetate. These are diffusible dyes that, when cleaved by redox reactive oxygen species, will liberate a fluorescent molecule, dichlorofluorocene, right? So in this, again, 3-component mix, what John did was he stained B. longum in this red dye so that you can study just this population in flow cytometry, and then it co-incubate and then add this DCFDA. Now, if you look at the flow plots here, you fish out again the red component, right? And then you ask the question, do they become green upon pressure by the accompanying microbes? And indeed, if you have the Blastocystis added to B. longum or the E. coli and Blastocystis, there's a shift to the more fluorescent component quadrant, indicating that there is oxidative stress on this population of bacteria. Right? Again, you can quantify it on histograms, and these are significant changes. So there's oxidative stress, and that is probably one way in which Bifidobacterium longum is being suppressed in the in vitro community. Now, the host is also obviously involved, and we know, and this is also John's basis of John's PhD work, that Blastocystis, when co-incubated or exposed to host epithelial cells in the gut actually produce antimicrobial peptides as a host response. So does Blastocystis, when inducing this generic innate response, does it have a negative impact on B. longum? So co-incubate Blastocystis to prime the monolayer of intestinal cells, and then remove Blastocystis and add B. longum and then count. So controls have a certain number, but for primed Blastocystis prime epithelial cells, they lose, they get fewer in number. So that is also a host effect through Blastocystis on Bifidobacterium longum. So in more elaborate experiments, and these are called barrier function studies, and we know that Blastocystis subtype 7 does compromise barrier function in host tissue, right? And there's this a very classical approach where you can look at electrical resistance across a membrane, and these should be high if the membrane is intact, but if there's a perforation or breach, then the resistance is lowered. And there's also movement of fluorescent molecules through breached barriers, right? So if there's a breach, there's a kind of a flux of fluorescent molecules through, and you can measure the lower compartment to look for increase in flux. So for Blastocystis, if you look at resistance, there's loss of resistance. But if you add Bifidobacterium longum, there is a recovery beyond control. So resistance is restored. And the story is also true for dextran-fit-C flux, right? So there's an increase in flux due to Blastocystis compromise of barrier function, but this flux is restored even beyond control. So the flux is blocked in the presence of Bifidobacterium longum. So Bifidobacterium longum, as has been appreciated and understood in the past, does strengthen the epithelial barrier in the gut. So that was all in vitro. And the second part of the study involved mouse experiments that was first established and published by my former postdoc Sitara Ajampur in PLOS ONE back in 2016. She just essentially established a model by which Blastocystis can be used to infect mice, right? And what was determined from this is that over the course of this infection, in controls of which, when you do a histopathology of the colon or cecum, you see a very healthy gut because there's not much difference. But in Blastocystis exposed colon and cecum of the mice, there's ulceration and loss of symmetry. So if you look at the pathology index, it does go up for Blastocystis infected mice, right? But importantly, what is the abundance of these representative bacteria in Blastocystis infected mice? So total bacteria by qPCR, not much change over the 3 days, be it control or subtype 7B or H. Not much change for Bacteroides, but interestingly, for Bifidobacterium, there is a significant decrease in abundance after 3 days. And in unusual reflection of the in vitro data, E. coli is higher, and Lactobacillus, interestingly, is also lower. So you do have a decrease in so-called beneficial bacteria and increase in some of the pathogenicity associated bacteria. Right. So in summary, and this is how the paper kind of came together, you have Blastocystis subtype 7, which is more Asian-centric among human infections in our part of the world. And Blastocystis can exert negative effects on probiotic Bifidobacterium longum. E. coli can do this too, but together they can exert this through ROS mechanisms. Now, Blastocystis can have negative impact on epithelial cells, resulting in the induction of host innate immune responses that can have negative impact on Bifidobacterium longum. But we know that B. longum can improve the health of the epithelial tissues in the gut. So, you know, the general consensus in the Blastocystis community right now is that Blastocystis is actually beneficial for you. But is subtype 7 a wolf in sheep's clothing? Is it one of these so-called good or bad parasites masquerading as a good one? And what about subtype 4? Is it really the angel that we think it is? So is it really that good in terms of promoting health in the host? And that is the basis of Deng Lei's question. So Deng Lei read John's paper when he was in China. He was somehow enchanted by it and had a different take. He then applied to come to my lab and say, I want to work on subtype 4. And I think that subtype 4 has the opposing effect, right? So this is some data from Deng Lei's work. I just make sure my timing is okay. Yeah. And so it's a bit more microbiomes and immunology focused and a bit more animal colonization focused, right? So what he did is in this part of the study, he induced illness in the mice, and this is a DSS which caused inflammation, right? And then across the time as they recover from inflammation, but they're still ill, he adds Blastocystis subtype 4 through oral garbage, right? He feeds them over a course of time, and then he studies recovery and various other attributes. Now, over time, after you induce DSS colitis, there will be weight loss, right? And then there'll be recovery as you let them recover with normal food and water. But oddly, if you have Blastocystis, you recover much faster, right? And if you look at disease activity index, you look at the phenotypes of disease like ruffled feathers, lack of appetite, and so on. This slowly goes back down upon recovery, but Blastocystis colonized mice tend to recover faster. If you look at the histopathology or colon length, they're all reflective of a quicker resolution of disease in the presence of a so-called parasite. Like, so you find that quite odd. Now, let's go back to the alpha and beta diversity that I was talking about at the start. Let's just look at the OTUs, right? So again, the control mice at the start of DSS and then, you know, towards recovery, they're the same. But even though they recover, there's loss of diversity in the colitis mice. Blastocystis prevents the drop. So Blastocystis has a protective effect. And this is also evident in the Shannon and Simpson plots. If you move on to beta diversity and you look at controls, which is in the light green here, and controls after day 21 recovery from colitis, there's a distinct compositional shift. But in the presence of Blastocystis, this is not, you don't see this shift. That means Blastocystis again has a protective effect, preventing a compositional shift in the gut microbiome, right? And here you can see again that there are certain bacteria that go up, or there's a certain increase in bacteria in the control group after 21 days, right? And I'll give you more resolution in the next slide. And what's interesting and in line with Blastocystis having a protective effect is that in the control mice with colitis, there's an increase in certain pathogenic groups like the Escherichia Shigella group, which is higher in the control mice versus the subtype 4 colonized mice. In the subtype 4 colonized mice, there is an increase in these short-chain fatty acid producers, and these are known to be anti-inflammatory inducing bacteria. So Clostridium, Alloprevotella, and so on seem to be higher in Blastocystis colonized mice. So one additional thing that Deng Lei did was to study the immune subsets and cytokine profiles of the immune cells in the lamina propria of the intestines, right? And in mice that are exposed to subtype 4, there's a higher levels of interleukin 4, 5, and 13. And these are classic cytokines of the Th2 response. And Th2 response is typically a parasitic response, but less inflammatory and damaging than a Th1 viral response, for example. Think of COVID-19, right? And think of a worm infection, very different. Now, if you look at IL-10, which is a very key marker of anti-inflammatory tolerogenic response, much higher in Blastocystis colonized mice, right? And IL-17, which is the opposite marker for IL-10, has not much difference, right? And again, if you look at the Th1 markers, gamma interferon and alpha TNF, there is no difference. So it's clearly a Th2 dominant response. So just let me summarize this part of the study. Interestingly and unexpectedly, at least to me, is that colonization improves recovery of mice treated for 7 days with colitis-inducing drug. These subtype 4 colonized mice showed increase in alpha diversity, increase in so-called good bacteria, good for short-chain fatty acid production, decrease in pathogenic bacteria, and then a very clear Th2 dominant phenotype, and no change in the Th1 phenotype. So subtype 4 is associated with beneficial microbiota changes, non-inflammatory phenotype, and improved recovery from colitis. And this is just a pictorial representation of Teng Lei's work. So I mentioned the right part of the work, which is just increase in diversity or lack of loss of diversity rather, increase in certain good bacteria, decrease in certain bacteria associated with pathology, and a Th2 response. We also did similar work in just normal mice, and there was similar trends towards the healthy phenotype. So this is just for fun, right? Is Blastocystis subtype 4 good for gut health, and could it replace lactobacillus in these probiotics? Obviously, the answer is we don't know, right? This is very fundamental work that needs more work to establish more translational components. So the overall summary is my today's talk is that the gut microbiome plays critical roles in health and disease, right? So increased microbiome richness and diversity generally linked to good health, but not all the time. Disease states are characterized by low diversity microbiomes. Very important aspect of any microbiome project is the bioinformatician, because you need to crunch the data and give useful outputs, right? Eukaryotes are understudied but emerging components of the gut microbiota, especially the single-cell eukaryotes, such as Blastocystis, are increasingly recognized as ecosystem engineers. That means they can actually engineer the gut microbiota ecosystem, although they are a minority. And so we extend Tito's study, and then Teng Lei's study, which I just described, has been submitted, is under consideration in Microbiome Journal. But I would like the community to think about the impact of these studies and what should be done next. What will be the next steps to better understand the role of SCEs in gut health and disease, right? So I'd like to end off by acknowledging two groups. The Subtype 7 study, of course, was led by John and supported by honor student Iran, but the immunology work was, a lot of the work was supported by Yong and Jinwen, who are immunologists. The Subtype 4 study was led by Teng Lei, supported by his co-sup in China, Prof. Ping, but we had also Prof. Gascoigne, Lucas to help a lot with the immunology, right? And my last slide is a plug on a COVID-19 course. So besides doing Blastocystis, I also teach on COVID-19. And several of you have attended our previous courses. And because COVID-19 has changed over the last year, we have a third installment of this course, which I can send to Windell the details later. We have a donor that makes this course free for everyone. It costs 3,000 Singapore dollars per participant. If you're interested, please join us. It starts in January next year. Right, so that's all I have for you today. Thank you for your time, and I'm happy to take any questions.